when we usually talk about how the human body works, we tend to fall back on this old industrial metaphor.
Right, the body is a machine.
Exactly. The body is a machine, food is the fuel, and vitamins are just, well, they're the oil keeping the gears from grinding.
Which is a very comforting image. I mean, it's simple.
It is,
Yeah.
but it completely underestimates what's actually happening at the cellular level. Because when you actually look closely at the biochemistry, vitamins are far more than just metabolic lubricants.
Oh, absolutely. They're incredibly complex coenzymes.
Yeah, and potent systemic hormones. I mean, they are direct modulators of human DNA. They aren't just oiling the machine, they are actively rewriting the software that runs the entire city.
Which is a huge paradigm shift for a lot of people.
It really is. So if you're a medical student, a young healthcare professional, or just, a dedicated self-taught bio nerd, you already know vitamins are essential. But today, we're doing a deep dive into the definitive Handbook of Vitamins, 4th edition.
Edited by Jano Simplani and his colleagues, yeah.
Right. And our mission today is to look at the microscopic reality of how these molecules actually command our cells without, getting bogged down in all the unnecessary jargon.
Okay, let's unpack this. We're going to start with the architects of the system, the fat-soluble vitamins. So that's A, D, E, and K.
That's a highly logical starting point, really, because if we want to understand vitamins, we have to look at their physical properties first.
Right, the fat-soluble part.
Exactly. They are lipophilic, they love fat, and because of this, they can easily cross the lipid membranes of our cells and actually penetrate right into the nucleus.
So they aren't just floating around in the blood.
No, not at all. Instead of just facilitating quick chemical reactions, their mechanisms often involve significant cellular storage and direct interaction with our genome.
So they act a lot more like systemic hormones than simple dietary supplements. Let's look at vitamin A as our first example, because in our diets, we basically encounter two main forms, right?
Yeah, primarily two. We ingest preformed vitamin A, so the retinoids, from animal products. And then we have provitamin A, the carotenoids like beta-carotene, which we get from plants. But our bodies can't just use a carrot's beta-carotene in its raw state.
Exactly. The human liver basically acts like a chemical refinery here. It takes those dietary precursors and bioactivates them. First, it converts them into a molecule called 11-cis-retinol.
Which is the vision one.
Right. It's absolutely crucial for your vision because it physically binds to a protein called opsin in your eye to, capture photons of light. But the refinery also creates a second, profoundly powerful molecule, all-trans-retinoic acid.
And this is where it stops acting like a simple vitamin and starts acting like a hormone. How does that actually work inside the cell?
It's a fascinating mechanism. So all-trans-retinoic acid enters the nucleus of the cell and literally regulates gene transcription.
Wow, by binding to receptors.
Exactly. It binds to specific nuclear receptors. Specifically, it forms this protein complex called an RRXR heterodimer.
RRXR heterodimer.
Yeah. And you can picture this complex as a molecular key. It physically latches onto your DNA helix and controls the differentiation of almost every cell in your body. It basically tells a stem cell whether to become a skin cell, an immune cell, or a gut cell.
That is wild.
Wow.
And because it's stored in the liver, clinical professionals actually have to measure how much is banked in there. The handbook mentions the relative dose response test.
The RDR test, yeah.
How does that tell us what's going on in the liver?
Well, it exploits the body's own transport system. The liver produces this transport vehicle called retinol binding protein, or ApoRBP.
Okay.
If a patient is malnourished and their liver is starved of vitamin A, this empty ApoRBP protein just builds up in the liver tissue. It's just sitting there waiting for retinol to bind to it.
Ah, I see.
So the RDR test essentially measures that backlog, which gives clinicians a highly accurate, functional picture of the patient's internal storage.
Which perfectly bridges us to vitamin D, because it's also acting as a nuclear commander. Now, we call it a vitamin, but structurally the handbook classifies it as a secosteroid.
Right, which just means the seco part 1 of its structural carbon rings, the B ring specifically, has naturally broken open.
Right. And we ingest it as D2, ergocalciferol from plants, or D3, gocalciferol from animal sources, plus whatever we synthesize in our skin from sunlight. But just like vitamin A, it needs to be refined.
Precisely. It undergoes a two-step bioactivation.
Liver first.
First the liver and then the kidneys. They hydroxylate it. They add oxygen and hydrogen atoms to create its active steroid hormone form, which is called 1-alpha-25 dihydroxyvitamin D3.
Catchy name.
Very catchy. But this active hormone then travels to the cell nucleus and binds to the vitamin D receptor, or VDR.
And its main job is bone health, right?
Yes, but more broadly, its primary directive across the entire body is maintaining calcium and phosphorus homeostasis in the blood.
Because without that exact elemental balance in the blood, bone integrity fails.
Exactly. And even more critically, your neuromuscular system cannot fire properly without that precise calcium level.
So vitamin D is regulating the macroscopic structure, the bones and the blood calcium. But who is protecting the microscopic structure of the cell itself?
That would be vitamin E.
Right. Now, there are several tocopherols and tocotrienols out there in nature, but the handbook is very clear that only one specific type actually meets human dietary requirements: alpha tocopherol.
Alpha tocopherol, yes.
Why is the body so incredibly picky about this?
It comes down to a highly selective bouncer in the liver.
A bouncer?
Yeah, the liver produces a specific transport protein called the hepatic alpha tocopherol transfer protein, alpha TTP.
Okay.
And this protein has an exclusive affinity for alpha tocopherol. It binds to it and ships it out into the bloodstream to be used. The other forms of vitamin E just, they aren't recognized by this transport protein.
So they basically just get excreted.
Exactly. But once alpha tocopherol is distributed throughout the body, it embeds itself into our cellular membranes. Its job is to act as a chain-breaking antioxidant.
And when we say chain breaking, we're talking about stopping lipid peroxidation.
Right. Free radicals try to steal electrons from the fats in our cell membranes.
Which creates a destructive chain reaction that can literally tear the cell wall apart.
Yeah, it's cellular destruction. But vitamin E acts as a biological shield. It sacrifices its own electrons to halt that destruction.
That's amazing. Which brings us to our final fat-soluble powerhouse, vitamin K. We get phylloquinone from leafy green plants, and then monoquinones are actually synthesized by the bacteria living right inside our own gut.
Which is a great symbiotic relationship. Biochemically, vitamin K serves as an essential cofactor for an enzyme called gamma-glutamyl carboxylase.
Right, and that specific enzyme is basically the absolute master switch for blood coagulation. It activates critical blood coagulation proteins, primarily prothrombin.
And bone-building proteins too, like osteocalcin.
Yeah. I always like to visualize vitamin K as a highly specialized mechanic working on a fleet of ambulances.
I like that analogy.
Yeah, so the factory, which is your liver, can build the ambulance. It can manufacture all these inactive clotting proteins. But until that mechanic, vitamin K, actually installs the siren and the flashing lights, which is the gamma carboxylation process, that ambulance cannot do its job in an emergency. The protein simply won't clot.
It's completely useless until K does its job.
Right. But hold on, if vitamin K is the fundamental trigger for blood clotting, how do common blood thinners like warfarin actually work? Are they just going in and destroying the patient's vitamin K?
What's fascinating here is the sheer elegance of the pharmacological intervention.
Okay.
Blood thinners like warfarin and decamerol, they do not destroy vitamin K directly. Instead, they act as metabolic roadblocks.
Roadblocks?
Yeah, they block a specific recycling enzyme called vitamin K epoxide reductase. You see, when vitamin K helps carboxylate a protein, when it installs that siren, it gets oxidized and loses its active state.
Oh, so it's a one-time use thing unless it gets recycled.
Exactly. To be used again, it has to be reduced or recycled by this reductase enzyme.
Oh, I see. So warfarin just blocks the recycling plant.
Exactly. By blocking that one recycling step, the body rapidly runs out of the active form of vitamin K. The mechanic goes on strike.
So the liver keeps pumping out inactive clotting proteins, but they never get activated.
Precisely. The entire systemic physiology of coagulation is altered just by inhibiting that single recycling pathway.
That is incredible. So the fat-soluble vitamins act as these long-term architects, membrane guardians, and emergency dispatchers. But the body also needs a rapid response team, right? Molecules that are burning through the metabolic assembly line every single millisecond.
Yes, the water-soluble vitamins.
Right, the B complex and vitamin C. Because they are water-soluble, they aren't stored well in tissues. They must be constantly replenished. Let's look at the energy metabolism group first.
This group is the literal engine of cellular respiration. We begin with thiamine, vitamin B1.
Okay, B1.
Its active form is thiamine pyrophosphate, or TPP. If you want to extract energy from carbohydrates, you absolutely require TPP.
It's a mandatory cofactor.
Right, for oxidative decarboxylation reactions. Most notably for the alpha-ketoglutarate dehydrogenase complex in the Krebs cycle.
So without thiamine, the Krebs cycle just grinds to a halt. Glucose cannot be fully metabolized into ATP.
Exactly. And clinically, this profound energy failure results in beriberi, which can manifest as, fatal heart failure in wet beriberi or severe neuropathy in dry beriberi.
Do we have riboflavin, vitamin B2? I like to think of B2 as the manufacturing facility for your cellular shuttle buses, the coenzymes FMN and FAD.
Shuttle bus is a great way to put it.
Yeah, their entire biochemical purpose is to pick up high-energy electrons from the food you eat and physically transport them to the mitochondria so your cells can breathe. And a fun trivia fact about riboflavin, it is intensely sensitive to UV light.
Oh, yeah. The milk bottles.
Yes, that's actually the exact historical reason why milk, which is super rich in B2, used to be delivered in opaque or colored glass bottles to stop the morning sunlight from physically destroying the vitamin before you even drank it.
That's a perfect example of biochemistry dictating everyday logistics.
Mhm.
Next is niacin, vitamin B3. Similar to riboflavin, niacin is the precursor to NAD and NADP, which are the ultimate electron carriers in redox reactions.
So when a patient is deficient in niacin, the cellular energy grid collapses because electrons literally cannot be moved.
Right. Historically, this caused pellagra, a disease infamous for the 4Ds, dermatitis, diarrhea, dementia, and death.
Because tissues with high cellular turnover, like the skin and the gut, they just fail first.
Exactly. And rounding out the energy team is pantothenic acid vitamin B5. This molecule forms the very core of coenzyme A, or CoA, which is the universal currency required for lipid metabolism and entering the Krebs cycle.
So that's the energy generation side. But parallel to the energy group, we have the amino acid and one carbon metabolism group,
Right, starting with vitamin B6, pyridoxine.
which is converted into pyridoxal 5-phosphate, or PLP.
Hmm.
The rule of thumb in biochemistry is if a reaction involves moving an amino acid, PLP is probably there.
No, almost always. It's essential for transamination, swapping amino groups around to build new proteins, and decarboxylation, which is exactly how our brains synthesize crucial neurotransmitters like serotonin and dopamine.
And we absolutely must highlight biotin. It is the essential cofactor for oxalase enzymes, such as acetyl-CoA carboxylase, which is the first step in manufacturing fatty acids.
And biotin is quite famous in medical trivia because of a protein called avidin.
In raw egg whites.
Yes. Avidin is found in raw egg whites, and it binds to biotin with such extreme affinity that if a person consumes large quantities of raw eggs, it will physically strip the biotin from their digestive tract.
Wow.
Yeah, and it creates a severe clinical deficiency.
Note to self, cook the eggs. Next are folate and vitamin B12, also known as cobalamin. These two are deeply biochemically intertwined in one-carbon transfers and nucleotide synthesis.
You literally need them to build the actual letters of your DNA.
Right. How exactly do they work together, and what makes B12 so unique?
Well, B12 is a structural marvel. It possesses this massive corin ring with a rare, heavy cobalt atom sitting right in the center.
Hence the name cobalamin.
Exactly. But its absorption is notoriously complex. It requires a specific glycoprotein called gastric intrinsic factor.
Which is secreted by the choroidal cells in the stomach.
Right. The intrinsic factor binds to B12 and basically escorts it to the small intestine for absorption. Without intrinsic factor, maybe due to an autoimmune condition, you simply cannot absorb B12.
Leading to pernicious anemia.
Exactly.
And clinically, deficiencies in either folate or B12 cause megaloblastic anemia. Because the cells are trying to divide, they bulk up their cytoplasm. But without folate and B12, the DNA can't replicate.
Right.
So you end up with these abnormally massive, dysfunctional red blood cells.
They also regulate homocysteine levels, and folate is famously critical in early pregnancy to facilitate the massive cellular division needed to close the neural tube.
Right, neural tube defects. Finally, in the water-soluble category, we have vitamin C, ascorbic acid. Structurally, it's just a simple lactone ring, but it's an incredibly powerful electron donor. Now, everyone knows it prevents scurvy, but the how is really fascinating.
It all comes down to collagen synthesis. Collagen is the primary structural protein in your body, and it's woven together like a three-stranded rope.
Okay.
To weave those strands, an enzyme called prolyl hydroxylase is required. However, this enzyme relies on an iron atom at its core. If that iron becomes oxidized, the enzyme stops working.
So where does vitamin C come in?
Vitamin C's highly specific role is to constantly donate electrons to that iron atom, keeping it in its reduced state. When vitamin C is absent, the iron oxidizes, the enzyme fails, and the collagen fibers cannot cross-link.
So the structural rope literally falls apart.
Yes, leading to the bleeding gums, loose teeth, and unhealed wounds that define scurvy in sailors.
Okay, here's a question that always fascinated me. If we look at the B vitamins, they are basically generalized metabolic factory workers, right? They are in every single cell of the body, helping process basic fuel. So why do their deficiencies cause such bizarre, highly specific neurological symptoms?
That's a great question.
For instance, why does a lack of thiamine cause the specific eye paralysis and ataxia seen in Wernicke-Korsakoff syndrome, rather than just a generalized body-wide fatigue?
Well, if we connect this to the broader physiological picture, it's all about tissue-specific demand and margin for error. The brain is incredibly greedy.
Greedy for energy.
Exactly. It accounts for about 20% of your body's total resting energy expenditure, despite being a tiny fraction of your body weight. And more importantly, unlike your muscles, which can burn fat, the brain relies almost entirely on glucose metabolism to survive.
And as we established, burning glucose is strictly dependent on thiamine and niacin.
Right. Furthermore, the brain requires constant rapid neurotransmitter synthesis to fire its synapses, which depends heavily on vitamin B6.
So it's basically a power grid issue.
Exactly. When the generalized assembly line slows down due to a water-soluble vitamin deficiency, the system with the absolute highest demand and the lowest margin for error, the nervous system, is simply the first to critically fail.
The neurons literally run out of energy and communication molecules before the rest of the body's tissues reach their breaking point.
That's exactly what happens.
The grid fails where the power demand is the highest. That makes total sense. Okay, we've covered classical biochemistry. Let's transition to segment 3, the cutting edge. We are stepping out of the 20th century and into 21st century molecular biology. We're looking at epigenetics and AMS analytics.
Which is where things get really exciting.
Because the handbook highlights that vitamins don't just facilitate chemical reactions, they literally physically alter our genome.
Right. So epigenetics is the study of how behaviors and environment can cause changes that affect the way your genes work. Unlike genetic mutations, epigenetic changes do not change your underlying DNA sequence.
They just change how it's read.
Exactly. They change how your cellular machinery reads that sequence. Think of your DNA as a library of blueprints. Epigenetics determines which blueprints are left open on the table and which ones are locked away in a drawer.
And vitamins are the molecular keys operating those locks.
Yes. They target the DNA directly, and they target the histones.
And histones are those protein spools that DNA wraps itself around to fit inside the nucleus, right? The handbook discusses 2 major modifications here. Biotenylation, which is the physical attachment of biotin to these histones, and poly-ADP reoscillation, which is driven by niacin.
Right. And when vitamins modify these histone spools, it changes the entire chromatin structure. It can wind the DNA so tight that the cellular machinery can't read it, essentially turning a gene off.
Or it can loosen the spool up so genes are actively expressed.
Exactly. And then you have the folate cycle, which is responsible for producing S-adenosylmethionine, or SAM.
SAM, the body's universal methyl donor.
Yes. SAM literally hands over chemical methyl groups to be attached directly to the DNA strand itself, a process called DNA methylation. This directly dictates whether specific genes, including vital tumor suppressor genes, are silenced or expressed.
It fundamentally changes our understanding of nutrition. You aren't just feeding your metabolism, you're feeding the exact machinery that dictates your gene expression.
It's incredible.
But to truly understand these pathways, scientists have to measure them with extreme precision in living humans. This is where accelerator mass spectrometry, or AMS, comes in. This is a revolutionary analytical tool.
It really is a game changer for nutrition.
Because in the past, to trace where a vitamin goes in a human body, you had to use massive pharmacological doses of radioactive isotopes, which isn't totally safe, and it certainly isn't reflective of normal everyday digestion.
Not at all.
But AMS allows researchers to trace radio-labeled vitamins like 14C folate or 14C beta-carotene in humans safely at what are called attomole levels.
And we really need to emphasize just how small an attomole is. An attomole is 1/quintillionth of a mole. It's a billionth of a billionth. It is a scale so incomprehensibly small that researchers are actually tracking everyday physiological dietary doses safely.
It's basically like attaching a microscopic molecular GPS tracker to a single vitamin. Scientists can literally watch a single carbon atom attached to a molecule of folate as it navigates the human body.
Yeah, they can see exactly how many minutes it spends in the liver, when it enters the bloodstream, and what its exact metabolic fate is in real time.
That's wild.
And if we connect this to the bigger picture, it bridges a massive gap between microscopic molecular biology and macroscopic public health.
Because for decades, Nutritional recommendations were based on rough estimates, or simply finding the threshold where deficiency diseases like scurvy appeared.
Exactly. AMS gives us the exact, real-time kinetic data of how nutrients are absorbed, stored, and depleted in healthy living human beings. This incredibly precise data is exactly what is required to establish accurate, scientifically sound dietary recommendations for the public.
Which is the perfect biological link into our final segment, clinical application and deciphering DRIs. We're moving from tracking a single atom in a lab to applying this knowledge to millions of people.
Right.
We use a clinical framework called dietary reference intakes, or DRIs. These replace the old recommended dietary allowances as the gold standard. Let's break down the acronyms for our clinical listeners. First, you have the EEA.
The estimated average requirement.
Right. This is the intake level mathematically estimated to meet the biological needs of exactly 50% of a healthy population.
Correct. And from that ER, we calculate the modern RDA, the recommended dietary allowance. Mathematically, it's derived by taking the ER and adding 2 standard deviations.
So by doing that, the RDA covers the nutritional needs of 97 to 98% of the population.
Exactly. But when there isn't quite enough rigid scientific data to confidently set an ER, the committee uses an AI, or adequate intake, which is a thoroughly observed estimate of what healthy people seem to need.
Finally, and crucially, in today's era of heavy over-the-counter supplementation, we have the UL, the tolerable upper intake level.
Yes, the UL is very important.
This is the absolute maximum daily dose of a vitamin you can take safely on a chronic basis before risking chemical toxicity. So, what does this all mean? If you are a medical professional planning a patient's diet or evaluating their lab charts, how do you actually apply these acronyms in practice?
It requires a strict shift in perspective based on who you are treating. The EE is a tool for evaluating the prevalence of inadequate intakes within groups or large populations.
Okay, so it's a public health metric.
Yes. But when you are sitting in an exam room across from a single patient, the RDA is your clinical target. It is the intake goal designed to ensure optimal health for an individual.
And as a clinician, you must always cross-reference their total daily intake, especially including those broad-spectrum supplements, against the UL to prevent hypervitaminosis.
Which can be just as dangerous as a deficiency, frankly.
Absolutely. So as a quick review exercise to solidify your learning from this deep dive, let's look at a clinical scenario based on everything we just covered. Imagine you have a patient on warfarin therapy to prevent blood clots. Suddenly, without consulting you, they begin taking a high-dose, broad-spectrum vitamin supplement.
A classic scenario.
Right. A few days later, their lab results show their blood is beginning to clot dangerously fast. Based on our deep dive into enzymatic mechanisms today, which specific vitamin in that supplement is responsible, what enzyme is it interacting with, and what is the biochemical workaround happening in the patient's liver?
Think back to our ambulance and mechanic analogy. The culprit is vitamin K. By taking high supplemental doses of vitamin K, the patient is physically bypassing the pharmacological roadblock.
Because warfarin stops the enzyme vitamin K epoxide reductase from recycling the used vitamin.
Exactly. But if you flood the liver with fresh, unoxidized vitamin K from a giant supplement pill, the primary enzyme gamma-glutamyl carboxylase doesn't need to wait for recycled molecules anymore.
It just grabs the fresh supply to carboxylate prothrombin.
Yes, kickstarting the coagulation cascade and completely overriding the warfarin medication.
It's pure biochemical supply and demand. You bypass the broken recycling plant by importing fresh parts. Okay, I want to leave you with one final provocative thought to mull over. We've talked about how vitamins physically modify DNA expression through epigenetics and how every individual has a slightly different metabolic rate.
Right.
We also know about single nucleotide polymorphisms, SNPs, which are tiny individual genetic variations that can drastically alter how fast or slow your unique body processes specific vitamins, like folate. With our advancing understanding of epigenetics and high-speed genomics, are we rapidly approaching an era of pharmacogenomic nutrition?
That is a fascinating concept.
Right, an era where the standard population-wide RDA becomes totally obsolete, and instead, every single patient receives a hyper-personalized DRI, a bespoke biochemical blueprint based entirely on their unique DNA sequence.
This raises a profound question about the future of medicine and dietetics. We really are moving away from treating the statistical population to treating the individual genome.
The blueprint is getting personal. Remember, the vitamins you ingest aren't just oiling the gears of your body. They are the master programmers constantly rewriting your cellular code. Thank you so much for joining us on this deep dive into the molecular world of vitamins. Stay curious.